Recent neuroscientific advances into how our brain evolves, especially when we're adults, are absolutely stunning. And we would like here to dive into this research together with Lara Boyd, who came all the way to us from Canada. Lara is a professor in the Department of Physical Therapy at the University of British Columbia. She's a neuroscientist and physical therapist. Dr. Boyd directs the Brain Behaviour Lab at the University of British Columbia, and her research is on how brain function relates to behavior during learning. She's an expert in neuroimaging and neurophysiology and uses a variety of cutting-edge technology in her research. Lara, welcome on stage.
So thank you for inviting me. I feel a little bit like a fish out of water, but I hope that the information I have to share with you will at least be interesting, might promote a little bit of your own brain health, and maybe there's a way to make money on it, although if you figure that out, let me know, because I've been thinking about it. But so far, I'm just a poor academic, and that's okay, too. So what I'm interested in is how do we learn? And specifically, why are some of us better at learning than others? And so what my research tries to do is to try to seek basic knowledge about learning and then leverage that such that we can develop approaches and innovative technologies that will allow us to stimulate learning, learning faster, learning in terms of recovery after brain injury or insult. And we're also particularly interested in how we can take this information and help children who struggle during learning to allow them to become more functional and healthier and more contributing adults in society. By the way, this is my brain in the image here. And what I'm actually showing you is a picture, it's a wiring diagram of all the axons, all the long brain cells connecting all the different areas of my brain. So neuroscience has just undergone a fantastic revolution in the last 20 years. And I've been just lucky enough, honestly, to be in a front row seat for it. So 20 years ago, we thought that the brain was static, that after probably adolescence, it never changed, and that there was not much we could do other than hurt it. And in fact, we thought that from birth, you were really losing neurons throughout your lifetime. So we used to think we were over the hill when we were 30. Until we turned 30, then we pushed it to 40. And now, of course, at least in my view, you're not over the hill till you're 60 or 70 at least, because I can't possibly be there yet. But it turns out that we actually are probably not adding neurons to our brain, probably after about age 11 or 12. And so we've all been over the hill for a great deal of time now. We just didn't actually know it. So with that knowledge, though, with the knowledge that we're losing brain cells throughout our life, how is it that we are able to learn, to acquire a new skill, to develop the expertise and the innovation that's really represented in this room to push ourselves forward? And it turns out that in the last 20 years, what we've discovered is that our brains are constantly reorganizing themselves. And they're doing this in response to every activity, every behavior, every new fact or event we engage. And it's really a nonstop process. And this is what we mean by this term neuroplasticity, or plasticity in the brain. So anytime you learn something new, you engage in a new behavior. You engage in many behaviors, even sedentary behavior, which we're all practicing very nicely right now. You're actually changing your brain. And the exciting part about this, and also the scary part about it, is that you're doing this constantly. So it's not something that just children do. We're doing it throughout our lifespan. So we're always changing our brain, and we're changing it for better. We can also be changing it for worse. And it depends very much upon how we decide to engage in different activities and behaviors, the healthy or the unhealthy lifestyle choices we make, how we're pushing that brain around. But it's highly responsive and highly dynamic. And in fact, you're doing it right now, and I love making the point that when you leave this room at the end of today, your brain will be vastly different than it was when you entered the room this morning. So there's three basic processes by which you're changing your brain, and these happen both independently and they interact, and it tells us a lot about how we can shift our behavior to optimize brain health and optimize learning if that's really what we're choosing to do. So we can change our brain chemistry, we can change our brain structure, and then we can change how regions of the brain are functioning together. And these probably operate or largely operate together, but I'm going to talk about them in isolation just to make a couple points about how and why learning looks the way it does. So the first I'll talk to you about is brain chemistry. And I'm going to back up just a tiny bit and tell you a little bit about just how the brain looks, what the architecture of the brain is like, because it kind of helps make sense of how it can change. So if you look at the image up here on your left, this is an electron microscope view actually of neurons, of brain cells. And they remind me very much of trees. It's an analogy I like to use. So you're thinking about a very dense forest of trees. You have these long trunks of the tree, which we call axons. They're a part of the nerve cell that's really the long wire connecting brain regions. These can be very, very long. So one example of that is a neuron in my motor area of my brain. If I wiggle my first finger, it goes all the way down to my spinal cord. So quite a long distance before it makes its first connection. So those are the connections, that's the wiring. We also have the branches of the tree, or what are called dendrites in the brain. So this is where we have the projections off the top of the neuron that come very close to, just approximate the connection of the neuron next door. And those are very important when we think about the changes in brain chemistry. So when we practice a skill or we learn something new, the fastest and the easiest thing to change in the brain is the amount of chemicals that are between those connections at the very tippy ends of this tree, between these dendrites. It's called a synapse. There's a very small junction there. Actually, information is transmitted from one neuron to the next by chemical transmission across these small synaptic clefts. So as soon as you start practicing something, or if you learn something new, the very first thing that happens is you change the concentration of those chemicals. And that enables the neurons to respond more quickly the next time they're needed to transmit information across that synaptic cleft and activate the neuron next door. Now we can actually image this in humans using something called magnetic resonance spectroscopy. That's what the image on the left-hand side behind me is showing. These are actually individuals with stroke, and we're able to show that their neurochemical concentrates are vastly different, vastly reduced as compared to a healthy, age-matched individual. And it's actually something that allows us to produce a medical target for a drug or an activity that can change that. So the second way that your brain can change when you're learning is by altering its structure. So I told you that we don't really add new neurons, but we can change brain structure in other ways. We can add more of these connecting dendrites out on the periphery, or we can reduce these connecting dendrites. So we can have, if you add connecting dendrites and then you add chemicals to them, you can see now we get a much larger response during a behavior. So behaviors that are very well refined, that were very well practiced, this may be one method by which that occurs. Now, what's interesting about changes in brain structure is that they take time. They don't happen very quickly. They also probably take sleep. And this is why sleep is such an important element of actually learning, is because you have to give your brain the opportunity to change its brain structure so that you can have this difference in neuronal response. So you can think about it this way. If you combine changes in brain structure and chemistry, they explain why we have short-term memories and long-term memories. So we've all had the experience of practicing something, right? Practicing, say we're practicing a piece on piano. We practice, and we think, oh, I've got it. I can do this. I've got it down. You come back the next day, and you totally don't have it. Something has happened overnight, or actually not happened overnight, that failed to convert that short-term memory into a long-term. So these changes in short-term memory, they're supported by chemical increases in the brain. Changes in long-term memory have to have a structural change coming behind that chemical increase. That process in between, it's called consolidation. It's where we take a long-term memory, we take a short-term memory, we convert it into our long-term memory system so we can have it later, so we can access it later on. And that's one point where memories are very fragile. Well, learning can be disrupted. It also provides us an opportunity, a point where we can intervene to try to push memories towards becoming permanent so we can access them later if we want to. And I'll circle back to that later in my talk when I tell you a little bit about a couple of the interventions that we've been developing to try to speed long-term memory. So the last way that your brain can change to support learning is by changing its function. So this is just by having different brain regions come on together in concert with one another or very precisely timed in a sequence in order to support learning. And this is very easily measured in our lab or in many labs around the world using something called functional magnetic resonance imaging. It gives us these bright spots on the brain, which are brain activity associated with a task. An important thing to know about brain function is that your brain is actually never at rest. So even when you're at rest, you're seemingly doing nothing, and your brain continues to be highly active. And when we're at rest and we don't think we're doing anything, and our mind is actually wandering, and some of you may be there right now, your brain goes to a very stereotypic pattern of activity, something we call the default mode network. We all do it when we go to a rest state. When we allow our brain to just wander, we're seemingly off task. And I know that we often catch ourselves in that state and think, oh, I've got to get myself back together. I have these things to take care of. I have work to do, and I'm off task. But it turns out that allowing your brain to go to default mode network, allowing your brain to take a break and actually rest and wander may be an incredibly healthy thing for you to do for your mind. It may actually be very healthy for creativity, for spontaneously creative thoughts and experiences. And there's actually some very kind of rising concern among neuroscientists that we never allow ourselves to go to this state anymore, to just let our mind wander. And that's because of technology, which I would have called disruptive, but I heard the last talk. But what do we do if we're bored and we have really nothing to do and we're sitting still? We all take out that computer in our pocket and we start looking at it. And it may not be your best health decision that you make in your day. It may be just fine to let your mind wander, to be off task, and to just give your brain a little bit of a break for a moment. We think it may reflect a functional resetting of the mind. And this may be one of the reasons why different meditative therapies, perhaps even the mindfulness that all of us are more and more interested in these days, may show some significant health benefits. So I just described to you that the brain is massively neuroplastic. We're neuroplastic throughout the lifespan. Neuroplasticity probably supports recovery after things like brain injury and stroke. We know that we're doing it all the time, whether or not we want to. So this is a question that I fall on every day in my lab, and that is, if our brains are so amazingly neuroplastic, they're so capable of change, why can't I learn anything I want? Why can't I be fluent in French? Why sometimes do kids fail in school? Do they fail to take advantage of being able to just learn for 6 hours a day? What a magnificent experience in hindsight that is. And why don't people fully get better after brain damage? So I study stroke in my lab, and I can promise you, people do very poorly after a certain initial amount of recovery. We don't see the brain being able to adapt very well to those types of insults. So we become very interested in, well, what's the limit on neuroplasticity? Why can't we make it do what we want? Why can't we push it to counter these different changes? So one of the things about neuroplasticity is that it's what we call experience dependent. So the adaptive capacity of your brain is most influenced by your behavior. So there's absolutely no drug that stimulates neuroplastic change. And trust me, it's not for lack of trying. This is something that, of course, Big Pharma would love to solve, to make us all these magnificent learners. Changing behavior means changing brain. And one of the things we have to be aware of is that that can be for the better, but it can also be for the worse. So there's many things that we can engage in that are very negative for our brain, and those will also shape our brain in another direction. Now, one of the things that we've looked at in terms of learning is how do we help people have the right experience during learning to really optimize their brain change and optimize their change in behavior. That's really the goal. And so one of the things that we've found is that it's actually very, very helpful for both brain and behavior if learning is hard. So we say that struggle during learning is good. And we had an experiment we published about 2 years ago where this was very, very evident. So we had actually a motor task that people were learning. It's a semi-immersive virtual reality game where people have a very important job. They have to catch an asteroid before it hits the Earth and blows it up and kills us all. And people become very invested in this, and it's a pretty fun game. What we found was that if that task is too hard, people can't catch the asteroid, we don't see very much change in their brain. And if it's too easy, we don't see very much change in their brain. But when we make it just hard enough, that's when we really see optimized neuroplasticity, optimized change in behavior. And so there seems to be this sweet spot for learning. We call this the Goldilocks experiment. It can't be too cold, it can't be too hot. It's got to be just right. Now the trick is that that sweet spot is not the same for everyone. And this is also a theme that comes back to us over and over again in my research, and that is there's no one-size-fits-all solution in terms of learning. Our brains are all massively unique. The behavioral experience we bring to every task, every learning environment is massively unique. And those are the factors that become very tricky to account for. So in our work, what we're trying to do is develop algorithms, mathematics, that can adjust for many, many, many different individual factors and then try to adjust the learning experience to really optimize learning. And I think there's some really huge implications for education when you think about learning in that sphere and thinking about how can we make it quite unique for the learner rather than having every fourth grader learn multiplication in the winter term, which is kind of what happens now. There's no accounting for, is that fourth grader's brain ready? Was it ready? Was their brain maybe ready 2 years ago and now you're just wasting their time? Or maybe their brain won't be ready for another year and you're setting them up to fail. So not all neuroplastic change is positive. We see things like repetitive use injuries, chronic pain is an example of negative neuroplastic change. Your brain is now misinterpreting a pain signal as being much larger than it really is. Addiction, whether it's to drug, alcohol, coffee. In Canada, we're about to legalize cannabis. Any of those things, those are also going to change your brain. And the one I thought I'd highlight really quickly here, because I think it's one we all get to live with quite a bit, is stress and anxiety. And this can also shift your brain to become more negatively neuroplastic or inducing changes that we don't really want. So when we're highly stressed, we release a hormone called cortisol. And cortisol is actually not all bad. It has a bad rap. So we release cortisol normally. So all of us woke up this morning, which is good. It's always a good thing, whether or not it feels good. And we woke up because our brains actually released a little bit of cortisol, and it aroused us. So cortisol helps us be motivated. It helps us wake up in the morning. And if we have too little cortisol, we suffer from low motivation, low arousal. We're very sleepy. Now, if we're chronically stressed and anxious, though, we can keep cortisol higher than it needs to be and higher for a long period of time. So cortisol normally gets released and then decreases through the day. Finally, it gets to a level so low that we can go to sleep at the end of the day. Now, if we are too stressed, cortisol will be released in the morning and will just persistently stay high. And there's a number of negative side effects to persistently high cortisol. One is that we develop anxiety. A second is that we have health issues, so it promotes higher cholesterol, it promotes inflammation, lower immunity, weight gain, specifically abdominal weight gain. And in particular, it actually has been shown to impair our memory and impair our ability to form new memories. So this elevated cortisol, it actually competes with a chemical in our brain. It's called a trophic factor or a growth factor. And specifically, the one that it competes with is something called brain-derived neurotrophic factor. So early on, when I was telling you about changes in chemistry and structure in the brain, I didn't tell you that there are these growth factors in the brain that enable it to be more plastic, enable the structural changes to occur more easily, enable chemical concentrations to change more rapidly. And brain-derived neurotrophic factor is one of them. So if we have high cholesterol, we have low brain-derived neurotrophic factor. And we then become less able to learn new things, less able to remember things we already knew. And we actually see that over the long term, we'll see atrophy or a shrinking of key memory structures in the brain as a result of this persistently high cortisol. Sorry, cholesterol is bad too, but that's a different talk. So how do we manage this? So there is some very good data of ways that we can manage cortisol. We live in a high stress environment. All of us are probably fairly busy, high achieving people. So how do we counter this? So one that's very effective is Buddhist meditation, or meditation in general. So you see significant decreases in cortisol and blood pressure over a 6-week course or class of engaging in meditation. Did anybody in this room sleep 8 hours last night? A couple. Good on you. I've been up since 3, so I definitely don't fit into this category. So my cortisol is certainly elevated, which is probably why I just said cholesterol. But if you simply increase your sleep from 6 to 8 hours, you reduce the amount of circulating stress hormone, cortisol, in your bloodstream by 50%. And that's a very, very simple thing to do. You also significantly advantage yourself in terms of learning, because I remember I told you that sleep is actually somehow a key element in the changes in your brain and brain structure that are accompanying learning. And this is a big mystery in neuroscience, by the way. We don't know why we need to sleep. There's lots of theories. We think we're clearing out waste from the brain and something called the glymphatic system. We know that if you don't sleep, you very rapidly become psychotic. It takes only about 2 or 3 days. You can ask any new mom about that, and she can relate her experiences to you. But we're not sure exactly biologically why sleep is so key. But we do know that when we don't sleep, lots of very bad things happen. Lastly, exercise. And I'm going to shift and talk a little bit about exercise as I start to wrap up here. When we initially start exercise, our cortisol goes up, but it rapidly rebounds and actually in the long term is lowered by exercise. And so simply engaging in fairly moderate intensity exercise on a regular basis, even in a high stress environment, is a very, very good way and a very effective low-cost way of keeping your cortisol lowered. So when we think about learning, there's one other problem I want to highlight, and then I want to talk about maybe some solutions. So when we're trying to learn something new, it turns out that it often takes what we call, at least in my group, a high dose of practice to learn it. So you can think about practice of learning something new as just a dose, like you would the dose of a drug. And so when we look at animal work and we look at laboratory-based learning, we're trying to say change in behavior and change in brain, and we look at these doses, we find these huge numbers, like on my slide. So the middle bullet point there, that's some of the work from my group, where we see that it takes about 10,000 movements before we're really good at something. And this comes back to this old idea that it's 10,000 hours of practice before you're really an expert at something. It's hard to put an exact number on it. 10,000 is probably a bit of an exaggeration in terms of hours of practice, but it's a big number. And so this is a big problem. Many of us just simply don't have the time or the resources to engage in this kind of dose of practice. And certainly if we circle back to the example of our kids trying to learn in school, we certainly don't have school systems that are set up to enable kids to practice with this magnitude. So in my group, we've taken an approach of thinking about what kind of augmented technology or intervention could we use to prepare the brain to learn. So we think about priming the brain, getting it really ready to take advantage of new information so that you don't need as much practice and you get the same outcome. And we've looked at robotics, and we're looking at robotics. We have non-invasive brain stimulation where we actually can interact with the brain and actually change the excitability of certain regions of the brain prior to practice. And then we're looking very closely at exercise, and that's the one I'll just talk to you about today. And I'm happy to talk with anyone about the other two, robotics or brain stimulation, offline or at another time. So the intervention that I'm going to talk to you about really briefly now is 9 minutes of cycling exercise. That's it. We do 3 times 3 minutes of exercise. There's a tiny catch coming, and that is that people are going at 90% of their maximal capacity in this exercise bout. So it's a total of 9 minutes of exercise at near failure, so almost as hard as you can possibly go. And if any of you have ever done an exercise test, you'll know that this is the point right before you basically spit off the end of the treadmill and fly across the room because you simply can't go anymore. So the way that we've done these studies is we have people engage in this bout of exercise, and then try to learn something new. And then we test them 24 hours later to indeed see if they've learned something new and whether or not they've changed their brain. So why exercise? Well, we know that it does lots of nice things for the brain. It increases blood flow. It increases the numbers of vessels, blood vessels formed in the brain. We see changes in the structure of the brain in different regions. It facilitates the growth of synapses. It increases the amount of brain-derived neurotrophic factor, decreases cortisol, and increases neurotransmitters. And so what I'm showing you here, these are two neurons on the side. One is in a mouse that has not exercised, or from a mouse, because it's a from situation. And the other one is in a mouse that has engaged in chronic exercise. And you just see a much more rich and elaborate brain cell or neuron. So these actually are our data, and we've done this experiment in many different ways, published it. And I'll just walk you through this really quickly. I hope it's not too geeky. I think you'll find it interesting. So in our experiment, we do two things to see if someone's learning. This is a movement experiment. We have people practice movements that are repeated. If you improve on a repeated movement, we say you've learned. We also have people practice movements that are random. You can get better at random movements, but they don't show learning. They just show improved motor control or movement potential. So we have individuals who've exercised, and they're in the black. And we have individuals who've just had a period of rest. They actually watch a Planet Earth video for a few minutes, the same amount of time as the exercisers, just to maintain attention. And so here's our rest group in gray. So this is the day where they practice. So people exercise, and then they perform this task that they're learning. And we see no difference between the exercise and the non-exercise groups. So if we'd stopped our experiment here, we would have said exercise has no effect on learning. We have instead people come back 24 hours later. There's no exercise on the second day. They just come in and they go right to work. And this is what we see. We see a significant advantage for the group who've exercised. And in fact, if you compare their performance here to here, they've learned overnight. It's called offline learning. They've gone home, they've had the rest of their day, had dinner, slept. We don't control any of those behaviors. And they come back the next day and they've learned overnight. They've improved overnight. And so now we see a very significant advantage of exercise. We see it's very specific to the repeated or the learned sequence rather than to the random sequence. This one is not different. And so this is a method by which we've shown in many different experiments now we can actually facilitate more learning given the same dose of practice. Now, when we start to unpack it, we see a number of different things, but the one I'll highlight to you here is that we see this large increase in brain-derived neurotrophic factor after exercise. And so we think this is one of the mechanisms by which the exercise is having a positive effect on learning. Now, I promised you that one size did not fit all, and I don't want to disappoint. So one other thing we've done with this work is consider genetic influences. And just to show that I'm really not that smart at all, when we started off this work, there's actually a gene that codes for brain-derived neurotrophic factor. And we were quite confident that the expression of this gene would tell us whether or not someone was an exercise responder. That turned out not at all to be the case. Instead, we also looked at some other genes, and this is a gene that codes for dopamine. Now, you may know dopamine. It's the neurotransmitter that is depleted in people with Parkinson's disease, and it's very important for learning. Now, some people express a type of dopamine gene where the two copies of the gene are identical, and in about 30% of the population, the two copies of the gene are not identical. These people are called heterozygotes. So they don't express dopamine quite as well as the other 70% of the population. And it turns out that the expression of this dopamine gene tells us whether or not exercise is the right intervention to stimulate learning. So in people who express the two same copies, they're called the homozygotes of the dopamine gene, they have an excellent response to exercise. And the people who exercise but are the heterozygotes, they do not show the same effect. So they actually don't benefit from exercise. We might have done other good things to them, but we didn't help their learning. So this brings us to this last idea then, and you may have heard, and you heard actually earlier in one of the pitches about this idea of personalizing medicine. So collecting a lot of data on individuals and then tailoring their individual health experience. So I'm going to push that out a little bit farther, and what we're thinking about is personalized learning. So how do we understand the individual factors in each person, each child, each patient, that we're trying to help learn and then tailor their learning experience best for their particular brain. And it's kind of a massive problem because we have to account for all of their past behaviors that have shaped the brain to that point. Probably we have to account for their genetics. And then we also have to design an intervention that's appropriate for them and their capacity, so we have that particular difficulty being at the right point. But I think that's an exercise that's going to only yield benefit because the whole world is better when we have engaged learners, we have a smart population, we have people recovering better from brain damage. So for you, as you walk out of this room, I guess I would encourage you to engage in the behaviors that you want because they're all going to shape your brain. So choose very wisely how you spend your time, what you choose to put in your body, what you choose to practice. If you want to change your brain, practice is the very best way that you can possibly do that. And just remember that the uniqueness of your particular brain are going to shape you as a learner and also as a teacher of learned behaviors as you encounter and go through your daily life. So with that, I would thank you, and you're all welcome to visit beautiful Vancouver anytime you're up. It did look like that today, by the way.